Hey guys, how you doing? It's me again. So you guys are currently watching some videos on natural selection that are put up by the College Board, which I think are going to be hugely helpful. There were a couple pieces of information that we missed from the last section, or Unit 6, and they had to do with like DNA technology and organism development. So the DNA technology piece, a lot of the things that we're able to do with DNA, manipulate genes, comes from our understanding of viruses and bacteria. So there's going to be some terminology that kind of pops up throughout, and hopefully I'll have some videos and what have you to kind of help support this a little bit. And we'll look at a few, not today, but we'll look at a few multiple choice and free response questions that kind of go along with that as well. Okay, just a little quick technology glitch there. Let's see if it works now. Oh, there we go. Awesome. Okay, so one of the things that we talk about is DNA cloning. So this is used a ton in order to make proteins. So let's typically we'll use something like insulin. Insulin is a protein that humans need to take sugar from their bloodstream and get it into their cells. So people who are diabetic, they don't produce enough insulin, and it's because in their pancreas, where the insulin is produced, the DNA is either has mutated, is defective in some way, that either doesn't allow them to make the right type of insulin or enough insulin. So one of the things that we used to do is we would take the pancreas or insulin from pigs. So as pigs were slaughtered, one of the things that they would save off would be the pancreas. They could extract or remove chemically the insulin from there, and then those that could be stored in a kind of cool place because it's made of protein. It only had a shelf life of a few weeks, and then people could take that pig insulin that had been cleaned and what have you, and inject that into themselves when they needed more insulin. Obviously, that it's nice to have insulin in a little pen or some type of device that you can stick yourself with, that doesn't have to be refrigerated. So over the course of time, one of the things they were able to do is they were able to make synthetic insulin, and they were able to do that using cloning of DNA. They could take the DNA sequence from humans that allows you to make DNA, give it to a bacteria. The bacteria would then take that, make DNA, RNA, and make proteins, and now you get this target protein or the protein that you want. And once again, we've used the mechanisms that are found inside of prokaryotes or bacteria, single-celled organisms, in order to benefit ourselves. Pretty neat little. So target protein. So it could be we can take this same strategy of having bacteria produce proteins and use it in a lot of different ways. We can use it with pesticides. We can use it with to digest toxic waste. So we can engineer new genes. So now that we've not only taken an existing gene, we can take our own. We can make our own DNA sequence and give this DNA sequence to bacteria to have them produce a protein that then does something we want it to do. Maybe, for example, we know there's a protein that helps to break down, like an enzymatic protein that helps break down certain types of toxic waste. For example, they've done this with oil spills, where they take bacteria that have been genetically modified and they'll spray that bacteria on the oil. And what will happen is the bacteria will eat or digest the oil and produce things that are not toxic. So this is another thing that we can do. We can also use it to create hormones for people. In addition to things like insulin, we can any proteins that we might need to produce for people. We can do this. All right. In the current COVID-19 thing that's going on, we know that there are antibodies, proteins that are found in the blood of people who have they called convalesced, meaning that people who are sick and get healthy again, they learn to develop these proteins that then help them fight off the infection later. We could do the same thing with those proteins. We could take those proteins and reverse engineer them and then have a bacteria produce the proteins that can then be mass produced in large quantities, which is super cool. Looks like we're going to have another issue here. Go in and out again. Okay, so wrong way, sorry. Let's see here. Okay. So another thing we can do is RFLP stands for restriction fragment length polymorphism. So another term for it is gel electrophoresis. What we're able to do is we know that so viruses infect things. They can infect things like humans, so they can get into our cells, drop off their DNA, can go through the lytic cycle, which is where the DNA is copied into RNA or copied into more DNA, and viruses make new virus particles that then are released from our cells. There's the lysogenic cycle, which is when the DNA from the virus gets added or inserted into the DNA of the host. All right. So in this case, this is where we're talking about in this process, raw DNA is cut. This is kind of where this kind of comes in. So let's say you're a bacteria and you're infected by a virus, and there are two types of viruses out there. There's lytic viruses that come in and destroy the host, and there's lysogenic viruses. Lysogenic viruses would take their DNA and they would add it into the host DNA. What some hosts have been able to do, so some bacteria, is develop enzymes called restriction enzymes that cut out the DNA of the viral infector. So let's say I'm a bacteria, I have this virus that lands on me. Virus lands on me, injects its DNA inside of me, and leaves. That DNA now can go through the lytic cycle, create more viruses, which it doesn't do in this case. Let's say it adds itself to the inside of my own DNA, my own genetic code. When this happens, now when I make a copy of myself, there'll be two of me. Each would have the viral infection built inside the DNA. How do we get rid of that? Well, we have no mechanism. However, bacteria have created this mechanism of restriction enzymes that allow them their special tools, special proteins that then look for the sequence of the viral DNA, cuts it out, and removes it. All right. We can use that same sequences. So let's say, for example, you guys heard of palindromes like taco, cat, race car, dad, mom, poop. They're all palindromes. They're read forward and backwards the same as far as words. So the way restriction enzymes work as well, they look for sequences of DNA that are read the same way forwards and back. So we know all of our restriction enzymes that we know about all come from bacteria. So bacteria have developed enzymes to then cut out viral DNA. So if we know what sequence it cuts for, we can take that restriction enzyme. I can take a long piece of DNA from, let's say, a human, and if I drop that restriction enzyme inside with the DNA, what's going to happen is it's going to try to find anywhere it finds the code, the secret AGCT code that's a palindrome, and it's going to cut the DNA into little pieces. So this is supposed to be a little video here, which doesn't seem to want to load. And here's another one that doesn't want to load either. We'll have to figure those out. But with these so this is our DNA. What the restriction enzyme is going to do is here's this protein. It's going to cut the DNA into small segments. So what I could do is I could take, let's say, two people. Let's say you have me, and we'll use Jeff Lamb as the example. He's one of the last people I talked to this morning. So if Jeff Lamb and I were convicted of a crime, all right, we both left some skin cells behind. They could take our DNA from our skin cells. They can put a restriction enzyme in with our DNA. All right. The restriction enzyme are going to cut the DNA based on our A's, G's, C's, and T's. It's going to look for certain sequences. Because Jeff Lamb and I are not the same person, all right, even if we were twins, we're still not the same person. We'd have genetic differences in our DNA. His cuts or his fragments, his cut DNA are going to be different than the DNA of myself. So I'm going to pull up a real quick video and show you. And let me just pause this for a sec. Pause for a sec. Okay, so we're back at it. So I found a quick little video I want to show you. I'm actually going to narrate the video instead of the narrator here. So, okay, so I want to talk about agarose gel electrophoresis. So agarose, OSE, obviously is a sugar. So, so let's go back to the restriction enzymes. So let's say we have a piece of DNA that's 100,000 bases long. Okay, so A's, G's, C's, and T's are the bases, and there's 100,000 of those. Let's say it's a gene from me, and it's a gene from Jeff Lamb. and I want to see which one of us committed a crime, let's say. I could take a gene that is not highly conserved. So in other words, a gene that there's a lot of mutations in. All right, so if Jeff and I both carry the gene, let's say it's a gene for, I don't know, skin color, which is a gene that's not highly conserved. I can take that gene and I can cut it into little pieces with a restriction enzyme. What the restriction enzyme does is it looks for A's, G's, C's, and T's, and when it finds a certain sequence, it makes a little cut, so it makes a fragment. So let's say there were 5 sequences of A's, G's, C's, and T's that the restriction enzyme liked. All right. If it's my piece of DNA that's 100,000 bases long, and there are 5 cuts, if I cut it 5 times, it gets 6 pieces. Let's say I take Jeff's. Let's say in Jeff's DNA, his had less mutations than mine. Maybe there were only 3 places that it cut. So instead of getting 5 cuts, there are 3 cuts. He would get 4 pieces. So there are these fragments that are produced. Hence the name restriction fragment polymorphism, or gel electrophoresis, right? That's that restriction fragment like polymorphism. So let's hit play. We actually have these devices at school. I wish we were there to actually do this right now. So typically what we'd have here is you would have a couple of devices here. You have a mini centrifuge, which these little tiny mini test tubes go in. You have a power supply. Power supply takes electricity and runs into this plastic box. And there are these things called pipettes, and the pipettes are to deliver small amounts of liquid into these little holes that you'll see in a sec. So let's let this kind of do its thing. So this is a gel reaction box, and so what happens here is in this gel reaction box, notice there's hopefully you're thinking about a car right now. There's a negative and positive terminal. Then you look at your car, you have a battery. There's a black cable and there's a red cable. The black is negative, and the red is positive. We have a black wire here and a red wire there, negative and positive. So electrophoresis, excuse me, we're going to take this gel of sugar and we're going to run electricity through it. And phoresis means to separate. So we're going to separate the fragments we created, my 6 fragments and Jeff's 4 fragments, into, I'm going to separate those fragments by size. So the way this is going to work is you would create a gel. All right, so they're showing you here are these electrical. This is where the electricity comes in. So the negative electricity comes here, the positive comes in there. They're showing this arrow here. This is the direction that the DNA is going to move, and the reason why the DNA will move in this direction is because DNA is negatively charged. So we're going to put that in, and here's our positive end. So I'm going to pause this for a sec here. So notice there's like this little tray, all right? If you've ever made Jello before, Jello, you take some type of powder, you add water to it, you boil it, and then when you let it cool, it solidifies. What they've done here is they have created this little slab of Jello, and this slab of Jello has tiny holes in it, right, called wells. And these wells are where we're going to put the DNA. So we'll talk about where the DNA comes from and how we get it after. That's a whole other video. So we're going to take this little gel, and the gel is going to sit inside. You can kind of see the holes in the gel right here. This looks like it has about 8 or 9 holes in it. So the neat thing about the gel, like if you've ever made Jello before, right? We'll go back. It's the same concept, right? So if I make Jello and I take whatever the powder is, and it says add 1 liter of water to the powder and boil it, great. I get Jello that's just the right consistency. If I add 2 liters of water instead of 1 liter of water, I get Jello that's really kind of watery or thin. If I only add half a liter of water, I get Jello that's a little bit more solid or a little bit more rigid. The same thing is true about this gel here. Depending on the consistency that you mix it, how much powder to how much liquid will give you either gel that's really thin or spacious, liquidy, or gel that's more compact. Depending on the size of the fragment you're dealing with, I said 100,000, right? There's a certain formula that you would mix a certain amount of powder to a certain amount of water to create this gel. So the gel, once the gel is solidified, it goes inside. All right. We want to be able to run electricity through it, so we're going to put, this is called a buffer. So a buffer is it's a solution that is going to allow the electricity to run through. Usually it's water with some type of like sodium bicarbonate, like baking soda in it, basically. All right, and what's going to happen is we're going to flood this up to the max level. All right, and let's go back a little bit. Let's go back a little bit more. Okay. So now what's going to happen is the electricity is going to be able to run from the negative end to the positive end. If you noticed on there, these are our DNA samples, right? So these are the samples that have had the DNA put inside, and they take a restriction enzyme and they use the same enzyme for each DNA sample. All right, this is that pipette that you were seeing earlier. The pipette allows you to pick up little tiny amounts of DNA. So there's the DNA in the liquid sample right there. So they're able to use this pipette, pick up a tiny amount of it, and what's going to happen is this sample, in addition to having DNA, it's got a stain or a dye in it that sticks to the DNA. It also has a chemical called glycerin, and glycerin is like heavier than water. So they're going to take this DNA sample, right? They have whatever this is measured in microliters, really small amount, and they're going to drop that DNA sample into those tiny little holes that were in the gel. Now, it's important to notice that this is the negative end, so the holes are on the negative end. And the reason why is because DNA is negative. So if I put a negative charge here and DNA is negative, and DNA is inside these little wells, it's going to be pushed away from the negative charge, and it's going to get pushed through the gel. And as it travels through the gel, depending on the thickness of the gel, the smallest pieces will travel the furthest, and the largest pieces will travel the shortest distance. This is kind of a nice little device. It's actually got the numbers. It actually numbers the wells here, which is kind of probably on the plastic below. It's kind of neat. So they drop the DNA inside. Now that tip, they'll get rid of that tip. They'll get a new tip, and they'll do the same thing in the third well for the third sample. And they'll do it again with fourth and fifth sample. So once again, basically what's happening here is they're taking DNA and putting DNA in each one of these wells. So let's say, let me go back just a little bit. Go right there. Whoops. Okay. So let's say that this is the crime scene DNA, and you had four. In this case, with just Jeff and I, you have person number one, person number two, but it looks like they're going to throw two more samples in there. So if this is the DNA found at the crime scene, and they have 1, 2, 3, 4 suspects, they can take the same enzyme, cut the DNA from the crime scene, cut the DNA from each of the suspects, and what's going to happen is it's going to cut differently because of depending on what gene they're looking at and what restriction enzyme they use. So the next piece of this is now we need to run our gel. And to run a gel, notice that here's the negative side, here's the positive side. The DNA is negative, so the electrical charge, and there's a cover that goes on top. The idea of this cover is it keeps you from electrocuting yourself. So this cover is gonna snap onto these posts. The negative electricity will run in here. It'll run through the gel and to the positive side. You'll see little bubbles on either side start to form. Okay. It uses just whole household current. So this is going to put 120 volts of electricity, give or take, through this, which obviously is not a good thing. Electricity and water don't really mix well. So as the electricity goes through, what's going to happen is here's the power supply. Negative goes to negative, red to red, black to black. Okay. It allows you to turn it on, change the voltage, change the amperage, power switch. The ones we have aren't quite as sophisticated as this, but they do the same basic thing. So here they're currently 0 volts. This allows them to increase the voltage. Looks like you're going to use 100 volts. And it's 400 milliamps. And this one is actually way nicer than ours. It has a time on it as well too. It's going to run it for 60 minutes and then shut it off. And then hit run. And here's the electricity running. So now there's electricity running through this liquid. As the electricity runs through the liquid, what's going to happen is it's going to cause the DNA that's inside these little wells to start to move. I'm going to show you. Okay, so notice here are the wells. Right. This is a really nice setup. It's got these measurements here, so you can actually measure the distance that things travel. It's got the wells numbered 1 through 8. What's going to happen now is the DNA is going to be pushed along with the stain, because remember the stain is stuck to the DNA, is going to get pushed through the gel. As it moves through the gel, the smaller pieces of DNA move further, and the larger pieces of DNA get stuck inside the gel. they get stuck inside the gel because the thickness of the gel does not allow those pieces to move. We can use this process of electrophoresis for any type of molecule that we know the charge of. For example, if it was a positively charged molecule, we would take the device and flip it, and wells would start on this side and get pushed towards a negative. So we can also do this for proteins as well as other molecules. Excuse me. All right. Now, one thing I want to show you here, towards the end, you start to see a differentiation of, in other words, there's no more gel here, or no more DNA here. It's all is moved. What we would look for in this, we would look for these bandings or these lines. Let me pull up a picture real quick and show you.